Hi, it's Miss Barnhart, and today we're going to talk about several techniques that are used by scientists today in biotechnology. These include restriction enzymes, vectors, gel electrophoresis, labeling, PCR, gene libraries, DNA sequencing, and microarrays. First, let's talk about restriction enzymes. These are also known as endonucleases, and they naturally occur in bacteria. Bacteria can be attacked by viruses, and these enzymes cut the foreign DNA into pieces so the bacteria can't be harmed by the virus. That's great for bacteria, but what's pretty cool is that scientists have figured out how to use this information to manipulate DNA. So, how do they work? Generally speaking, they recognize palindromic DNA sequences. These are sequences that read the same way from the 5' to 3' of each strand. They can either cut the DNA in the middle of a sequence, leaving blunt ends, or they can produce a 5' overhang of a few bases called sticky ends, as seen here. Restriction enzymes, or endonucleases, only cut the DNA at what we call restriction sites. These specific sequences of DNA are usually 4 to 8 base pairs long. You can think of them as DNA scissors. Literally hundreds of restriction enzymes have been isolated. One such example of a restriction enzyme is EcoR1. This recognizes the restriction site GAATTC, and it cuts between the G and the A on both strands, leaving a 4-base sticky end. Each endonuclease cuts at a different restriction sequence. These allow scientists to combine DNA from different sources as long as the DNA has been cut by the same restriction enzyme. That means they'll have the same sticky ends, so the base pairs will match up and they'll hydrogen bond with each other. Ligase, another enzyme, seals the break. This graphic shows how this is done. Restriction enzymes cut both the human gene and a bacterial plasmid with the same restriction enzyme. This allows them to join together and make recombinant DNA. When inserted into a bacterium, the bacteria can then make human proteins like insulin or human growth hormone. Next, let's talk about vectors. Vectors are DNA molecules that are modified to store and replicate other DNA sequences. Examples are bacterial plasmids, phages, viruses, and artificial chromosomes. You can see the plasmid, a small circular piece of DNA in the bacterium pictured here, as well as a phage, a virus that infects a bacterium. So how do these work? Well, to be useful, a plasmid must minimally have an origin of replication, a region with many restriction sites, a gene or genes that allow for screening or selection of cells that have taken up the plasmid, usually a gene that conveys antibiotic resistance. You can see in this map of a plasmid all of these features. However, there is a size limit on how much DNA we can insert into a plasmid. To get more genes into a cell, we must use artificial chromosomes. In 2010, the J. Craig Venter Institute created the first cell with a completely synthetic chromosome. Our next technique is gel electrophoresis. Gel electrophoresis is a technique that uses electricity to separate DNA fragments based upon size. So how does it work? First, DNA is placed into a matrix gel made of agarose. When the gel is exposed to an electric field, the DNA moves towards the positive electrode because DNA is negative with the phosphates. Different sized fragments move through the gel at different speeds. Smaller fragments move faster or further. A DNA profile, or DNA fingerprint, is a unique banding pattern of a specific restriction enzyme digest of a particular DNA sequence. Here is an image of how this works. DNA is mixed with a particular restriction enzyme and is placed into the well A. It's then covered with a buffer solution. When exposed to an electric current, the different length fragments travel at different speeds. When stained, you get the distinct banding pattern shown here at B. The next technique we will look at is labeling. What is it? Labeling is a way to identify or locate DNA sequences of interest. Here's how it works. DNA strands are denatured or separated. The fragments are exposed to a short strand of nucleotides which have been tagged with radioactive or fluorescent dyes. The probes bind to complementary sequences on the original DNA. This image shows how this occurs. Can you see how the complementary nature of DNA allows this to happen? Next, let's take a look at PCR, the polymerase chain reaction. PCR is a way to produce many copies of DNA without the use of cells. And it only creates copies of the target sequence of DNA. Here's how it works. The DNA sequence of interest is analyzed. Primers that will complementarily bind to both sides of the sequence are created. The DNA primers attack polymerase, which is one that can withstand high temperatures, and new nucleotides are placed into a machine called a thermocycler. After 30 cycles or replications, over 1 billion copies of the target sequence are produced. This graphic shows the basic process of heating to denature, then annealing primers at cooler temperatures, then extending the nucleotide chain. Each cycle doubles the number of DNA molecules formed. 2 cycles are shown here. Now let's take a look at gene libraries. These are gene collections stored in vectors. Here's how they work. The large sequences are cut into smaller segments. These smaller pieces are placed into vectors, and then the vectors are placed into cells. We then have these genes stored in the vectors. Here's a graphic illustrating the process. DNA sequencing is our next technique. This allows us to determine the order of bases in a sequence of DNA. Here's how this works. DNA is replicated in the presence of dideoxynucleotides, or DDNTs. When dideoxynucleotides are incorporated into a DNA strand, it terminates replication. The resulting strands of DNA are separated by gel electrophoresis and then read, either manually or automatically, and the sequence of DNA is then known. You can see on the graphic here how the modified nucleotide stops replication. Now let's look at microarrays. Microarrays are a way to visualize simultaneous gene expression for multiple genes. Here's how this works. The microarray contains a complementary sequence for different mRNAs on thousands of these microscopic spots. When you take the messenger RNA from a cell and expose it to the microarray, each cell that has a sequence complementary to the messenger RNA fluoresces. So then you analyze the fluorescence pattern. Here's a gene chip that contains the microarray. The next picture shows how the complementary sequences are housed on the chip. When exposed to the messenger RNA that's been transcribed, each fluorescent spot indicates that messenger RNA has been produced. Thus, we can see what genes are actually active in the cell using this process. In this lesson, we've examined a number of different techniques that have allowed us to better analyze and manipulate DNA. These are restriction enzymes, vectors, gel electrophoresis, labeling, PCR, gene libraries, gene sequencing, and microarrays. There's a lot of information here, so be sure to take a second or third look if necessary. I hope it's helpful.